Every human life begins as a biological blank slate, a single, unspecialized cell known as a zygote, carrying the full instructional manual for a new organism. To create this blank slate, biology requires two of the most hyperspecialized and distinct cells in the body, the sperm and the egg. Success depends on a multi-stage protocol where timing, chemistry, and spatial positioning must align with mathematical precision. This process systematically dismantles those specialized identities, using their genetic material to build the undifferentiated foundation of a brand new human life. The environment for this protocol is the ampulla, located in the outer third of the uterine tube. These source notes reveal the extreme attrition rate of the journey. Of the 200 to 500 million sperm deposited, only a few hundred survivors, between 300 and 500, actually reach the site of fertilization. These few survivors face a narrow window of opportunity, as the ovum remains viable for only 24 to 48 hours after ovulation. Even arrival does not guarantee entry. The sperm must first complete capacitation, a mandatory 7-hour biochemical priming process that allows them to interact with the egg. The journey is a race against a biological countdown, requiring absolute chemical synchronization before the window of viability closes. The surviving sperm immediately collide with the egg's first defensive barrier, which is a thick layer of follicular cells. They initiate the acrosome reaction, releasing the enzyme hyaluronidase to digest through this outer shell, known as the corona radiata. This technical breakdown illustrates the sequential barriers the sperm must navigate, from the corona radiata to the zona pellucida and finally the vitelline membrane. The sperm utilize lysosomal enzymes to trigger the zona reaction, disintegrating the second barrier to gain access to the egg's interior. Each step of this chemical sequence is highly specific, evolved to allow the sperm to navigate the egg's unique molecular defenses. The protocol reaches its final physical hurdle when the sperm contacts the vitelline membrane, using integrin peptides to initiate the fusion of the two cell membranes. Contact between the two membranes triggers an immediate, massive calcium wave that ripples across the entire spherical surface of the ovum. This calcium surge initiates the cortical reaction, which instantly hardens the egg shell to block all other sperm and prevent lethal polyspermy. The same wave provides the signal for the egg to finally resume and complete its second meiotic denission. The ovum maintains its genetic integrity by sealing itself off the moment the first breach occurs, permanently ending the competition for entry. Once the gates are closed, the egg pulls both the head and the tail of the sperm into its cytoplasm. As noted in the source material, this entry allows the male and female pronuclei to form, marking the start of the final phase of fusion. These two distinct nuclei migrate through the fluid, drifting toward each other until they seamlessly meld into a single entity. This merging is the true completion of the fertilization protocol, permanently erasing the individual identities of the original cells. This fusion results in the restoration of the full diploid number of chromosomes, providing the complete genetic set required for human life. At this precise moment, the chromosomal sex of the future individual is permanently established. By combining unique traits from two separate parents, the process ensures the genetic diversity necessary for a unique human being. The result is the zygote, a single unspecialized cell holding the complete genetic blueprint for embryonic development. This 48-hour race through a cellular gauntlet has produced a blank slate, the starting point for a brand new human life.